Good afternoon, and thank you for the panel, and thank you for the organizing committee for having me to present some of my work under the supervision of Professor Lihadh Al-Gazali and Professor Bassam Ali at the College of Medicine and Health Sciences at the UAE. So, my talk today is going to highlight how the advances in pediatric genomics have helped us to reach to a diagnosis to many of the undiagnosed cases in neurodevelopmental disorders in the country. So I will highlight the importance of genomics in pediatrics in general, particularly in the UAE. And while I'm doing that, I will explain what is a rare disease and why they are very common, relatively common in the UAE, and how to reach a diagnosis of them, and also take some examples from our lab. So why we use genomics in pediatrics? Mainly, there is an estimated 7,000 rare diseases so far. A rare disease is defined as the disease that occurs in 1 per 10,000 patients. And most of these rare disorders are monogenic in nature. That means they are caused by a disorder in one gene. So the majority of these monogenic disorders, they manifest at the early ages of life in pediatric clinics. And studies show that many of them also, an example of that, around 1,500 genes, although Professor Verlos earlier said there may be less than that, but studies show that they are above 1,000 gene, let's say, according to how we study those. So a better understanding of the genetic basis of the disease will help us reach a better prognosis and provide more accurate management. And also it will help us in the surveillance and the genetic advice for the family and the counseling of the family. And it will also stimulate, especially when you publish those, it will stimulate more research into novel therapies, developing novel therapies. And it will also help the policymakers into providing better support. So in the UAE, we found that because of the high consanguinity, the prevalence of the birth defects ranks the 6th worldwide. And most of these are diagnosed in the early 5 years of life. And it is really the 4th most common cause of death in the UAE, which is quite high when you look into other populations as well. So there is also healthcare limitations that mainly due because although there is a lot of effort and a lot of work and resources are put into this, there is still no complete variome or a database to look into these rare variants, which will help a lot in collecting data and reaching a diagnosis. So in our lab, we look into these as case-by-case or family-based research or family-based investigation. I just want to bring this map because it always shows how consanguinity and the prevalence of genetic disorders and rare disorders would be higher in the region of the GCC in the Middle East compared to the other parts of the world, and that correlates with the percentage or the prevalence of genetic disorders as well. The approach mainly have been looking into either the traditional genetic testing or the more modern, which is now traditional as well, using either whole exome sequencing or whole genome sequencing and microarray to look for the, to map the genome for the homozygous loci, or we can screen as single base or single gene or single locus by Sanger sequences, or less resolution than that using karyotyping. So I will go through some of the cases that we've been through. The first one is for a child, although we usually look into families with more than one affected child, but this actually showed, the pedigree shows high consanguinity for in different generations. So we suspected a genetic cause, an inherited cause directly, and we included the patient. So this child has progressive microcephaly and seizure disorder and intellectual disability. We've reviewed him when he was 3 years of age. And we found this null variant, which is, it's not very clear here, but it's a nonsense variant that will truncate the transcript of the POS3 gene. POS3 is a pseudo-uridine synthase gene that allows or catalyzes the pseudo-uridination of the transfer RNAs. And we found that this variant completely leads to the loss of the transcript this is a blood sample, RNA from the blood sample. So what we are looking now into, how would this gene would be involved in the intellectual disability and a severe neurodevelopmental phenotype. And early results show that it leads to defect in the proliferation of yeast that lack this in a knockout model, and we're looking into cellular models as well to prove our hypothesis. Now, the next gene is the SLC1A4, not 2, which is a serine, tyrosine, and cysteine transporter. The loss of that gene, the loss of function mutations in these genes would lead to a progressive microcephaly, spastic phenotype, and seizure disorder early on in life. What is interesting about this case is that this particular phenotype was associated to a particular population group. It was first diagnosed in Ashkenazi Jewish population, but our work showed that it might appear in other populations as well. The THSD1 loss of function, this is a family with 5 affected members with not intellectual disability, but or a neurodevelopmental phenotype. They had a form of non-immune hydrops fetalis early on, and it will progressively clear out, and they show skin disorder. Let's say, and what our work showed that because this gene was first associated with early embryonic lethality, but this family had evidence that this gene can be associated with a less severe form of the disease. What we did is that we found an 8 nucleotide deletion in the sequence of the in exon 4 of this gene, and that led to the truncation of the whole protein. This is a prediction, but the the thrombospondin site, which is this is a transmembrane protein, and the site where it binds to the membrane itself is lost. And we anticipate that this is the cause why it leads to the phenotype. So the next one, this child with the syndrome, the pontocerebellar hypoplasia 9, which is famous for the loss of the, it has a very known feature on the MRI, where you will have shrunken pons and cerebellum and complete absence of corpus callosum. And what we found in this particular, we detected a missense variant in exon 12. However, this gene, there is many variants that have been discovered and reported before as we work, proving that this is the causative gene or not. We needed to do functional analysis for the particular variant, but we already know the function of the effect of the relationship between the loss of function in this gene and the phenotype. So the location of the variant itself is not at a specific active or important part in the protein. However, it's near to the active site. This is just a prediction. So we did an enzyme activity assay in order to detect or validate the functional loss of the protein due to the mutation, which was proven by the decreased activity of the mutated protein compared to the wild type. So Donson loss of function in this conference, actually the last time, our professor have picked up a picture, Professor Al-Ghazali, that was presented here, and because of the striking phenotype and the severe features that show that the fetus would show with severe microcephaly, micromelia syndrome, we went directly to screen if there is any variance in the gene Donson, which is the downstream neighbor of SON, a replisome component. And what we found is that we were able to detect a new variant that is linked to this specific phenotype. The gene is associated with microcephaly and intellectual disability, which is a milder syndrome, and there is a founding mutation, a specific variant that is linked to the complete, to this striking phenotype, but using Sanger sequencing, we were able to detect another variant that would show, that had led to the same phenotype. What we end up with is that the complete loss of the protein would lead to a phenotype, which is the microcephaly, micromelia, and the mild loss of it due to decreased function of the protein would lead to a minor phenotype. So to wrap it up, we think that to stay updated with the advances in clinical genomics would be beneficial to pair that with the work at the genetics clinics, because it will add to the diagnostic ability of the clinics and labs. Because rare variants and rare disorders are more frequent in the region, consanguinity counseling the risk of consanguinity in counseling is crucial. And it is important to identify and report rare disorders because they will enrich databases that will help other labs as well to improve their work and their diagnostic yield. And it will reflect to better diagnostics and pave the way to developing novel therapies. I would like to thank all my lab members. Professor Lihadh Al-Gazali have been our supervisor, as we said, the mother of the lab, and truly an important, a key person in our lab and personal mentor. Professor Bassam Ali also, he has been a very supportive and we still look forward to working ahead with him. I thank Ms. Annie Joan, Ms. Dana Mahairi, and Ms. Fida, Ms. Asil, and Ms. Prasita. I see her, Dr. Prasita, I see her around, Dr. Afif, Dr. Nisreen, and Ms. Nisreen as well, all the members who have worked and have been working with us. And we have a dynamic lab, people go and come back, but the work is going. Thank you very much.
Thank you.
Look forward to this.